All of this made me realize that while I do love paleontology and extinct animals, what I really love is anatomy and bones and skeletons. I gained an interest in how our understanding of these modern animals and their anatomy could help us understand the anatomy of extinct animals. Now we have been able to tell from our using our studies of modern animals, we learned something cool about the extinct ones, about how they breathe.
Welcome back to Fossil Crates Live. In this episode, I sit down with Dr. Jessie Atterholt, an anatomist and paleontologist whose work bridges living and extinct animals. Dr. Atterholt shares how she became an anatomist and paleontologist, and how studying animals alive today can help us understand animals from the fossil record, including how extinct species may have breathed. Watch to the end for a fascinating look at how anatomy helps bring ancient life back to focus. And if you haven't already, please subscribe to the Fossil Crates YouTube channel. It's completely free and helps us bring more real paleontology to more people.
Of course, we're paleontologists, so we want to know, what does this tell us about extinct animals? And in order to be able to answer that question, what do we need?
Hello, hello. It is high noon in Phoenix, Arizona, and various and sundry times across the world. I see we have folks from the other side of the world, literally in South Korea. Welcome once again for Paleo Portals. Today we have an extra special guest. They're all extra special, as you know, but this is Dr. Jessie Atterholt. And Dr. Atterholt works out of California, where she is an anatomy instructor, but she also has a passion for paleontology. And we're going to talk about the intersection of anatomy, paleontology, how she became a paleontologist, and just have a great hour and change. So, Jessie, if you don't mind, tell us a little bit about yourself, how you come to be in the paleontology sphere.
Well, if it's okay, Brian, I'm going to go ahead and start off. Hi, everyone. It's so nice to see your faces. I'm really happy to be here with you today. I'm going to start off by sharing my screen with my presentation because I put some slides on that topic at the beginning.
Right on.
All right. Can everybody see this?
I can indeed.
Okay, so I did prepare a presentation, or rather, I'm recycling one from a previous talk I gave on some part of my research program. So I do a lot of research that focuses on weird anatomy inside the vertebral canal in a group of animals called archosaurs. So I'll tell you a little bit about that today. But I will start with just talking a little about my own journey through my life becoming a paleontologist. So like many of you guys, I was really little when I was smitten with dinosaurs and paleontology, and I decided I wanted to pursue that as a career. I was really lucky. I grew up in a family that did a lot of traveling and camping and visiting state parks, national parks, especially in southwestern United States when I was young. And one time we visited a state park called Anza Borrego Desert State Park. You can see a map of it in the upper right-hand corner there. This is in very, very, very southeastern California. It's actually the largest state park in the state. It is gorgeous badlands and really, really cool, wonderful, important fossils from the Pleistocene. So we're talking mammoths and mastodons and giant ground sloths and ancient cheetahs and all kinds of stuff. So we visited this state park. And by the way, I'm Southern California born and raised. So it was relatively close to where I was living, only 3 hours away. And found out they had a volunteer society, paleontology society, which consisted of retirees. But nonetheless, I wrote a letter because this was the 90s. So I wrote a letter asking if I could come and join the activities of the Volunteer Society for one day. And this wonderful gentleman that you see here, Mr. George Jefferson, he was the state park paleontologist at the time. Not only did he allow me to come down and visit, but he invited me and my mother to join the program as volunteers. So here I am on my very first visit in my little baby Triceratops t-shirt. This is me, age, I think 9. And so from the age of 9 until I graduated high school, I went out to Anza Borrego about once a month. And I got to learn the basics of geology, paleontological fieldwork, anatomy, all kinds of cool stuff. This also led me down another rabbit hole. So as I mentioned, Anza Borrego has Pleistocene fossils. So that means a lot of those organisms are quite closely related to life on Earth today. And so we have a whole collection of what we call comparative specimens, which are skeletal preparations of modern animals that we can use to help us identify our fossil animals. And they asked for volunteers to help prepare these skeletons, which pretty much involved skinning and gutting roadkill, and skeletonizing it, usually by boiling down the dead animals after they were skinned and gutted. And for some reason, I was probably 10 or 11, and I decided I wanted to try this. And my mother said, okay, you go for it. You're going to come back crying in 2 minutes. And I loved it. I thought it was so much fun and amazing. So here is some photos of myself and another volunteer, Michael Gubrick. In the right-hand photo, we're boiling down a coyote skeleton. And in the left-hand photo, we're actually skinning and gutting a cheetah, which was a donation to the state park from the San Diego Zoo. So that was extremely cool.
Is that a leg? Sorry to interrupt. Is that a leg laying there right in front of you?
Yes, I am. Don't be sorry to interrupt. And I meant to say anyone should actually interrupt at any time. This is not a formal presentation. So yes, my colleague over here is working on the cheetah, which has neither a head nor tail at this point. And we've chopped off a leg. And yes, I am skinning a cheetah leg over here. Yes. That is exactly what I'm doing. So all of this made me realize that while I do love paleontology and extinct animals, what I really love is anatomy and bones and skeletons. So I started collecting bones and skeletons, and I would collect roadkill when we would be, hiking, or, I lived out in the middle of nowhere in the desert, so I had access somewhat regularly to dead animals.
Is that a tarsier or something in the black and white to the left of your knee?
No, that's actually a bobcat.
That skull down there, that's a big, wide ocular. Holy
yeah.
Yeah.
Cats they have big eyes. So here's a bobcat. This is a mountain lion. This is my cast of a manatee skull, because manatees were my favorite animal when I was this age. So I started collecting bones, and sometimes I would have my favorite thing when I was this age was my Skulls Unlimited catalog. If you're not familiar with Skulls Unlimited, highly recommend checking it out. They sell amazing real bones and skeletons, and also reproductions. So that was my birthday present this year, was this manatee skull. But I was collecting and skeletonizing my own collection as well. I had a colony of domestic beetles at one point, flesh-eating beetles. That's how I actually skeletonized. This is a rat skeleton and a mouse skeleton. So I prepared these with my dermestids. I had really wonderful, supportive, patient parents. I'm very lucky. So the collection grew until it is what you see behind me today. So I am coming to you from my home, but my background, my fake background, is a picture of my real office. So I still have all of these bones and dead things, and my collection has only grown with time. So this is kind of a small snippet of it, what you see behind me, and everything on the ground in this old picture I still have it. So it makes me happy. So I love anatomy, love paleontology. I was super fortunate. I went to the University of Pennsylvania for my undergraduate studies, and I was advised by Dr. Peter Dodson, who is a ceratopsian paleontologist. This is a Peter in this picture right here. But the extra cool thing about going to UPenn was that Peter was a professor in their veterinary school. And through him, I started taking real true anatomy courses. So I kind of graduated from skinning and gutting the dead things to doing an actual careful dissection and studying soft tissue anatomy and not just skeletal anatomy. So I got to dissect ostriches, and then this is a monitor lizard that we dissected during my time there. Just a couple of examples of some fun things we got to study. I worked with Peter and some of his graduate students. So that was amazing. I didn't lose my interest in paleontology. Rather, I gained an interest in how our understanding of these modern animals and their anatomy could help us understand the anatomy of extinct animals. So when I went to the University of California, Berkeley for my graduate studies, had become, the fate had led me to study birds at this point. I did some dabbling in Mesozoic bird paleontology when I was an undergraduate. So I was interested in birds. And so I studied modern birds and extinct birds, both their anatomy, and also osteohistology was another field that I got into. So chopping up bones and being able to understand things about the physiology and the growth of extinct animals from those chopped up bones. So that's what I did as a graduate student. And then I'll let you stare at these pretty pictures. Well, I didn't make a slide for this, but Dr. Curtis asked me to share with you guys my postgraduate career path because it wasn't necessarily traditional or smooth. So when I finished graduate school, I was kind of a little bit disenchanted with the world of academia and research. I had discovered I really loved teaching. So I ended up taking a job working as a biology teacher at a private high school in Southern California. And I stayed there for a few years and then ultimately had the opportunity to transition to where I am now, which is Western University of Health Sciences. So I work at a university. I get to teach human anatomy to medical students. So I get to do dissections and study anatomy and teach it as my job, which is the best thing in the world and makes me very happy. And I still get to continue my research in paleontology and comparative anatomy generally. So I'm really lucky. One of my best friends and greatest colleagues is this gentleman here, Dr. Matt Wadle. He works at Western U with me. And what we have in common is that we both study archosaurs. So he does a lot of work on sauropods. I do a lot of work on birds, but they both belong to this group called archosaurs, which gives us some interesting common ground for collaborative research. So the research I am going to talk about with you today is some of my collaborations with this gentleman here. Has Matt given a talk, Paleoportals talk?
He has indeed. A legendary talk. He's the record holder for longest meeting. And yeah, we chatted about a variety. As you know, Matt got going.
Why am I not surprised? So then some or maybe all of you are familiar with the delightful person who is Dr. Matt Wadle. So as you may be familiar with some of this, but just in case you're not, or in case a review is helpful, this is a cladogram or a scientific family tree that shows us this group of animals that we call archosaurs. So it includes dinosaurs, both the extinct ones and the still living ones, our modern birds. It also includes pterosaurs and crocodilians. So modern alligators, crocodiles, caimans, and all of their paleo relatives. So there is this, when we want to study extinct animals, we also want to look at their closest living relatives. So we have a fancy term for this. It's called the extant phylogenetic bracket. So this really just means looking at the closest living relatives of these extinct animals. And for dinosaurs and pterosaurs, the extant phylogenetic bracket is birds and crocodilians. So a lot of paleontologists you'll find study these two groups of animals. So for myself, I have tended to go the way of birds. So I know a lot about birds, kind of. I study them, and I have a lot of data on birds. So what I had was of interest to Dr. Wadle. So I've introduced you now to archosaurs. That's our study group. And our particular area of anatomical interest is what we call the vertebral canal. So this here you're seeing the vertebrae, a vertebra from several different animals, an alligator, a sauropod, and an albatross. the vertebral, so a vertebra is just one of the bones that we stack together to make what we would call our spinal column. So the vertebral canal is this big hole in the middle of every vertebra. And that's the space that's occupied in life by the spinal cord. So when you stack a bunch of vertebrae together, then it creates this bony tunnel that the spinal cord sits in. So it's nice and protected. So one of our areas of research has been looking into respiratory structures that also are inside the vertebral canal. So this is a CT scan of a cross-section of an ostrich neck. So all of the gray stuff around here, that's the muscle of the neck. And then the white, lighter colored gray is bone. So that's one of the vertebrae. It's a cervical vertebra or a neck vertebra. Then this is our vertebral canal, this hole, this ring. And the gray blob inside of the vertebral canal is the spinal cord. And the black stuff inside of there is air. And that air actually represents these weird respiratory structures. So let me tell you a little more first about the bird respiratory system and how birds breathe and what exactly these little things are that we're looking for. So this is a diagram showing the avian respiratory system. So we're seeing the lungs and the trachea in pink. And then we're seeing something called the air sacs and diverticula in green. So as mammals, we humans just have lungs and a trachea, and we breathe air in, and then we breathe air out, and that's how we get our oxygen. Birds are much more complicated. Instead of breathing the air in and out in that simple way, they kind of just end up breathing the air in. It eventually will come out the trachea, but it travels in one direction across the lungs. The lungs are small and they're kind of rigid. They don't expand and collapse when the bird breathes like they do in us. And the lungs are connected to this really elaborate system of what we call air sacs. So air sacs are very thin membranous sacs. It's almost kind of like a bunch of balloons that fill up the bird's body and are connected to these lungs. So when the bird breathes in, the air sacs will inflate. And when the bird breathes out, they deflate. And they go everywhere in the body. They fill up the body cavities. They can be found in between skin and muscle, in between muscle and muscle, and even entering into bone, which we'll talk about in a little bit. So this is just another view of what these structures look like. So we're seeing the lungs back here in pink. We see trachea extending out to the head or the windpipe. And then all the stuff in green is these air sacs and the tiny little things that branch from them that we call diverticula. This is a slide about how birds breathe. It's actually pretty complicated, so we'll just skip that for now to save ourselves a little bit of time. But if anyone's interested, I'm happy to come back to it later. So the air sac system will extend into the bird's skeleton, which helps make it really light, but able to stay super strong. And like I said, the air sacs, they go everywhere in the bird's body. So this is the CT scan to the middle part of the body of a baby ostrich. So this is a baby ostrich. So if we had a CT scan looking at it right about here, this is what it looks like. And all of the black stuff inside, so this gray layer represents the outer part of the body. All the black stuff in here represents air and parts of the respiratory system. So you can see how these things are just full of air. So much air in them, really, really expansive respiratory system. So when these parts of the respiratory system go into the bone, they have to make holes in the bone to get in there. And this introduces a topic that is called, or a subject we call pneumaticity. You may have heard about this from other paleontologists as well. It's something we're really excited about. So when we study something that's still alive, and then we want to be able to use it to help us understand an extinct animal, we have to look for clues. And these clues on the skeleton, because that's usually, not always, but usually all we have left of the extinct animals, right, is their bones. So we look for these clues, we call them osteological correlates. So osteological refers to the bone, and correlate means it's something on the bone that's correlated with or associated with a soft tissue structure. So these holes that the respiratory system leaves on the skeleton are called pneumatic foramina or pneumatic fossae if they don't go all the way through the bone. So that's just a fancy term that means an indentation or a hole that's created by the respiratory system on the bone. And we see them in modern birds. This is a few examples of what they look like. So they have a distinct appearance. But what's really cool is we also see these osteological correlates, these pneumatic foramina and fossae in extinct animals too. So we have some examples here in vertebrae of sauropod dinosaurs, some examples in theropod dinosaurs, and also some examples in pterosaurs. So all of these are little holes in the skeleton created by the respiratory system. So what that tells us is that these animals, which are all archosaurs, remember we talked about that earlier, all of these archosaurs have a similar type of respiratory system. So we now know, we have good evidence that some extinct dinosaurs and pterosaurs had a respiratory system like birds with this weird air sac system. And the reason they evolved this is because it's a really highly efficient system. The way they breathe, they're able to keep oxygenated air flowing over the lungs when they breathe in and when they breathe out. So it's really, really great. It's a way of getting lots of oxygen in your body instead of only when you breathe in, like us in mammals. So now we have been able to tell from using our studies of modern animals, we learned something cool about the extinct ones, about how they breathe. That's not my discovery, by the way. Many people who came before me discovered that. But this idea of an osteological correlate is going to be really important for us moving forward. So now let's get back to my study with Dr. Wadel. So this is a diagram of an ostrich neck vertebra or cervical vertebra. And we can see this right here is what we would call the vertebral body or centrum. So this is where each vertebra will connect to the next one. This bit here is called the vertebral arch. So here's our vertebral foramen. There's a spinal cord sitting inside of it. And here's our vertebral foramen, spinal cord, I pointed that out. You might notice a couple of extra holes. Neck vertebrae have extra holes that we call the transverse foramen. That's where some important blood vessels travel to get up into your brain, the transverse foramen. So this is a diagram of an ostrich cervical vertebra. And this is a real life ostrich cervical vertebra. This one's actually been cut in half so we can see what the inside looks like. So here we're coming back to these weird parts of the respiratory system that go into the vertebral canal. They get a special name. They're called paramedullary airways. So para means next to, and medullary means the spinal cord. So these are airways, parts of that air sac system that are next to the spinal cord, which is really, really weird that parts of the respiratory system just hang out next to the spinal cord. So all of this stuff that looks kind of honeycomb-like is a bone of the vertebra, and this is our vertebral canal. That squishy thing is a spinal cord, and these black spaces are paramedullary airways. So here's a color coding system I'm going to use to help you be able to see these structures in the next few slides. So we have spinal cord in yellow, and then I'm going to show you paramedullary airways as those green blobs in a green color. And here's a couple of other structures related to the respiratory system. So we have supravertebral diverticula. So these are more small airways that will connect up with our paramedullary airways. And also traveling through these transverse foramina, we have intertransverse diverticula. So they're in life, there's a big blood vessel in there, but in birds, we also get some more diverticula. So all of these diverticula are going to connect up with each other in different ways. What's really cool about these structures is that we can identify them very easily from a CT scan. So if we aren't able to dissect a bunch of birds to see them like this, but instead we have access to a bunch of CT scans, which I did because I collected them for my research when I was in graduate school, Now we're able to study these in lots of different birds just from CT scans because they show up so well. You're just looking for air inside of the vertebral canal or these little black blobs. So whenever you do a scientific study, it's important to understand what other researchers have said about whatever it is you're trying to study. And so we did a search in the published literature for other studies on paramedullary airways, and we found only 4 papers, just these 4 papers. Here's some figures from those 4 papers. And of these 4 papers, only 2 of them really focus on paramedullary airways. These 2 down here, they make very brief mention of them. So they're only described in detail in one paper from 1908 that talks about paramedullary airways in the pigeon, and one paper from 1953 that talks about paramedullary airways in the turkey. That's it. So not a lot was previously known. So if we look at a fancy scientific family tree or phylogeny of modern birds, and we think about what groups paramedullary airways had been described in, there's only two groups out of the whole family tree where these structures have been described previously or studied. And they have been described as diverticula that occurred dorsal to the spinal cord or above the spinal cord. So this is what that looks like in CT scans, with the spinal cord in yellow and paramedullary airways sitting above it, hanging out above it. In our study, we collected data from 28 species representing 18 major groups from this bird family tree. So we were able to describe paramedullary airways from many, many more birds, which was really exciting. And we found that paramedullary airways are present in many different birds. These are all the different birds that we found them in. The birds that you see in boxes are ones that are in the same small group together, the same family, so to speak. We found that paramedullary airways are absent in a small handful of birds. So in loons, in a grebe, and in woodpeckers. So this makes some sense. For some of these birds, grebes and loons have very, very little pneumaticity. So their air respiratory system does not go inside of their bones very much. And that's because they are aquatic birds that hunt in a way that is called pursuit diving. So they basically spend a lot of time under the surface of the water, swimming around, trying to catch fish, catch their prey. And it doesn't help them if they are extra buoyant because they have all that extra air inside of their bones. That would make it really hard to stay underwater, right? Like if you take a deep breath and try to dive underwater, then it's really hard because your body's trying to float more with all that air in your lungs. But if you let your breath out, then you'll sink when you're in a swimming pool, or sink more easily. So same thing with these birds. They don't have a very extensive air sac system to begin with because of the way that they hunt their prey. So it wasn't very surprising that we would not find paramedullary airways here. However, it is a mystery why we would not see paramedullary airways in woodpeckers. Kind of weird. They're not pursuit divers. They eat little bugs that they find in trees when they peck on them and make holes. So we don't know why they don't have them. And then things get even more complicated, because if we look back at our birds that do have paramedullary airways, we see 2 that are classified as pursuit divers that do have them. So cormorants and an aquatic bird called a murre, they also spend a good amount of time diving underwater and chasing their prey, just like grebes and loons, but they do have paramedullary airways. So whenever we find a weird anatomical structure, we always want to know, is it an adaptation? What's the purpose of this? And in this case, we unfortunately don't know yet. It's still a mystery, or maybe there is no purpose. So let's talk a little bit now about the anatomy of these structures, the way that they look. So here we're seeing them in an ostrich, and there are 2 tubes that are dorsal to or sitting above the spinal cord. And what you're going to see on the next few slides, it's one CT scan slice going through a vertebra. And I'm showing you the same picture right and left. And on the right, you're going to see the color-coded version. And on the left, is the non-color coded version, so you can see these things with your own eyes. So sometimes there are two tubes, two airways that sit on top of the spinal cord. Sometimes, like we see in this cute little bush tit here, a small little songbird, there are two tubes, but now instead of being just on top of the spinal cord, they're lateral to it, or rather on the sides of the spinal cord. Sometimes there are two tubes, but they actually sit ventral to or below the spinal cord. That's what we're seeing here in this exotic bird called a violet turaco. And then in the ostrich, we also sometimes saw it as three tubes that sit on top of the spinal cord. So things are starting to get weird. We went from two tubes just on top of the spinal cord to now two tubes that can be anywhere. But now we have three tubes. Wow. Oh, and guess what? In the parakeet, we saw four tubes. So four different little extensions of the respiratory system sitting in contact with the spinal cord. Sometimes we don't see little separate tubes at all. They all seem to merge together into just one blob of respiratory tissue inside of the vertebral canal. So here again in the ostrich, you'll notice we saw a lot of variation of different types of airways in the ostrich than the way that they looked. So here we're seeing just one roughly C-shaped tube that's sitting dorsal to or on top of the spinal cord. And here in a pelican, this was the weirdest, craziest one. We're seeing the spinal cord in yellow, and it's completely surrounded by paramedullary airways. Totally surrounded, totally jacketed in it. So that really blew our minds when we saw it. And here's another example of that. So this is within the neck of the pelican where we're seeing it. This is in vertebrae that are farther down the vertebral column in the synsacrum, again in this bird called the violet turaco. And here we're seeing not only do we have paramedullary airways completely surrounding the spinal cord, but now they've totally pneumatized that vertebra. So there's almost no more bone inside of the vertebra. We can see it better here. There's an outer shell of bone that's in the right shape. And then inside is nothing but air and a spinal cord floating within it. So really weird stuff that was completely unknown before our study. So we want to know as scientists what factors can explain. We just saw a lot of anatomical variation in these structures, right? Sometimes they're two tubes, sometimes it's one, sometimes it's four, sometimes they're on top of the spinal cord or below it or all around it. We saw variation. So what can explain this variation that we see? One thing we thought was that maybe body size could explain it. So here we're seeing very large paramedullary diverticula in the two largest bodied birds in our data set, an ostrich and a pelican. So definitely really, really big paramedullary airways there. But then we can also compare this turkey vulture with a bush tit, and you can see that the paramedullary airways are pretty similar in size relative to the vertebral canal and the spinal cord, even though the turkey vulture is much bigger than our cute, tiny little bush tit. So maybe body size isn't great for explaining that. Well, what about vertebral region? And by that, I mean, what do we see in the neck vertebrae, cervical vertebrae, or the back vertebrae, dorsal vertebrae, different parts of our spinal column? In the cervical vertebrae, the neck vertebrae, we observed paramedullary airways present in every single bird where they were present at all. So all 23 species in which paramedullary airways are present. Zero absences in the cervical region, unless they were absent everywhere. Then in the dorsal vertebrae, so our back vertebrae, we found paramedullary airways present in 14 out of 23 species and absent in 9. And here we're seeing an example in dorsal vertebrae in a green-cheeked conure, this cute little bird. When we move even further back in the spinal column to what we call the synsacrum, we found paramedullary airways present in only 5 species and absent in 18. So now we can see that they're becoming a lot more rare. They also become a lot smaller as we move back through the spinal column. So we can see them here in the synsacrum of a great horned owl, and you can see now they're becoming quite tiny, little itty bitty. And then finally, when we get to the tail vertebrae of a bird or the caudal vertebrae, we observed paramedullary airways present in only 3 species, so absent in almost all of these birds. And here we're seeing an example in a pelican, one of the 3 where they were actually present. What was weird and cool about the pelican, remember, this is one where we saw paramedullary airways completely surrounding the spinal cord, and that persists all the way out to the very tippy end of the tail. So even though the spinal cord now is really tiny, it's just that little yellow dot or that little white dot here, and the vertebral canal is also really tiny, we still see that the spinal cord is surrounded by paramedullary airways. So the black stuff around there. Really weird, really cool. We also hypothesized that maybe development can explain the differences we saw in paramedullary airways, because we did have CT scans from some little baby birds and from adult birds. So maybe that was causing some of the differences we were seeing. So this part is just a very preliminary portion of our study. But here we can compare paramedullary airways in the neck of an ostrich chick with the neck of an adult ostrich. And in the chick, they're only about 36% the cross-sectional area of the vertebral canal, whereas that goes up to 51% in the adult. So a pretty big difference there. And we see a similar difference here if we compare the great horned owl chick with the great horned owl adult. In the chick, they're about 33% the cross-sectional area of the vertebral canal. And in the adult, that goes up to 48%. So we need to do further quantitative analysis on this, but that does suggest that these structures are becoming larger as the animal grows and matures. So what can we conclude? What is explaining all of this weird anatomical variation in these structures? Well, one thing is phylogenetic position. So there's what we would call strong phylogenetic signal in the presence or absence of these structures. So when we had a family of birds that had them, all of the specimens, all of the species we were able to check had paramedullary airways. And if a family had them absent, then all the species we were able to check for that family had them absent. So that helps us explain if they're there or not there completely. Body size doesn't really seem to help us explain the variation we were seeing, but that's something that we're going to investigate in the future in a quantitative way. Vertebral region definitely correlates very strongly with variation that we saw. So we see the biggest and most consistently present paramedullary airways in the cervical vertebrae, the neck, and then they become smaller and more frequently absent as we move all the way out to the tail. So we're not totally sure why. One reason could perhaps be that the neck is a part of a bird's body that moves a lot, certainly much more than the back or the tail. So maybe it's helping to sort of cushion the spinal cord in part of the body that's moving. And also where we have restricted movement back here, a lot less movement, than we have small or absent paramedullary airways. So we don't know for sure yet. Those are just some of our ideas right now. And then, like I already said, it seems like development also explains some of the variation we see. So we have bigger paramedullary airways in older animals. So that's something else for us to explore in the future. So now we talked a lot about paramedullary airways in living birds, which is cool. We got to see that they are present way more than we ever knew, that they do all kinds of really weird things, like completely surround the spinal cord. But of course, we're paleontologists, so we want to know, what does this tell us about extinct animals? And in order to be able to answer that question, what do we need? We need an osteological correlate. We need something, a sign of these structures left on the bone. And fortunately, because this is part of our respiratory system, that means they can leave behind pneumatic foramina and fossae, just like we were talking about earlier in other parts of the skeleton. So if we see foraminae, pneumatic foraminae or fossae inside of the vertebral canal, which we can see here in an albatross vertebra, or we can see here in the CT scan of a pelican vertebra, that tells us that the animal must have had paramedullary airways, because that's the only respiratory structure that would be inside the vertebral canal that could create these. Another osteological correlate we observed is not so much a hole or an indentation, but rather a weird texturing to the bone inside of the vertebral canal. So you can see how the bone out here is smooth, but the bone inside in this pelican has this weird, lumpy, bumpy texture. So what do we see when we look at the fossil record? We see sometimes pneumatic foramina inside of the vertebral canal. So this is the CT scan of a vertebra from a sauropod dinosaur called Giraffatitan. And you can see this is the vertebral canal. And then we have a hole in the roof of the vertebral canal that leads to spaces inside of the vertebra. So right here, this represents our pneumatic foramen. So we know that this animal definitely had paramedullary diverticula. So super exciting evidence from a sauropod. We also see this is a vertebra of another sauropod dinosaur called Alamosaurus. This is from a juvenile Alamosaurus. So the vertebra has fallen apart into two chunks. And now we're looking at just one half of that vertebral canal. And we can see the weird texturing that I talked about that we saw as an example in the pelican vertebra a few slides ago. So the weird texturing tells us that this sauropod dinosaur also had paramedullary diverticula. What about not dinosaurs? We know that pterosaurs also had an avian-like respiratory system. Well, here's an example of these pneumatic foramina in the floor of back vertebrae from a pteranodon. So now we have evidence of paramedullary airways in a pteranodon, a pterosaur. And this is an extra cool example because the foramina are at the bottom of the vertebral canal, the floor of the canal. In our living bird data set, we only saw that in the pelican. And the pelican's the one where it was completely surrounding the spinal cord. So we don't know this for sure, but that's a little bit suggestive that maybe in pteranodon, like the pelican, not only were paramedullary airways present, but they were completely surrounding the spinal cord and that really, really weird anatomy that we saw. So some exciting, cool food for thought. So that's the end of the first example of weird anatomical stuff going on inside the vertebral canal that I wanted to share. I also was going to talk about some different topic, weird vertebral or weird ridges of bone that we see inside the vertebral canal, but perhaps we'll pause for a sec and just like take any questions and talk and discuss. And if we get to my other subject, that's great. And if we don't, that's great too.
So I have lots of questions, but I'll hold mine. Mine are, although I did notice that two of your birds that lack it have red eyes. So yeah, I'm not saying that there's a correlation or causation there, but it was very interesting to see the two. Keep going to the left a couple more. Yeah, your two there had bright red eyes and they didn't have it, just saying. So I'm going to scroll back to the top because there's a lot of questions in the chat. If you want to not share screen for a moment so we can see everybody flying around here. Does your work help us figure out the key anatomical changes in the skeletal morphology from theropods to birds? What can your work on Mesozoic birds tell us?
Oh, really good question. So I think the work that I've shared with you today, I would say instead of telling us about the evolution from theropods to birds, kind of is more informative in the opposite way, I think of it. So telling us about the anatomy of theropods and these extinct dinosaurs long before they were on their way to becoming birds. And it's so cool and so weird because so many of the adaptations we see in birds that we think are for flight, like having very hollow bones, right? Having the respiratory system go in the bones. That's not for flight originally, at least, because it evolved first in these really big dinosaurs that were not flying. So I think what I'm sharing today is actually giving us insight into the soft tissue anatomy therefore also by the physiology of these extinct dinosaurs. So using birds to understand the extinct dinosaurs.
And then along those lines, how does your work on histology help figure out things such as the placement of air sacs in sauropods and theropods?
Oh, that's such a great question. So, osteohistology, we know from pretty recent work, just within the past couple of years, that air sacs can leave behind signs of their presence on bone in the form of a special tissue called pneumosteum, and it is possible to identify from histological thin sections. I actually have not had the chance to section anything and look for pneumosteum yet. But I have reviewed papers on it and I've read the papers on it, and I think it's super exciting. And I have projects in the works that will be exploring that.
Awesome. One that struck my eye was, would Hesperornis and kin probably have similar adaptations and therefore little pneumaticity?
That is a really good question. So one thing I did not include in here, I should have, but I forgot to add the slide. Sorry, guys. Forgot to update it. So as you can see from this work, I'm really interested in what we can learn from extinct animals. And I showed you an example of these pneumatic diverticula in the vertebral canal in sauropods and in pterosaurs. But I'm also interested in looking in Mesozoic birds. So I don't know about Hesperornis, but I do know about Ichthyornis and another close relative called Janornis. So those are not as aquatic as, why am I losing my words? What were you just asking me about?
Hesperornis.
Hesperornis, thank you. They're not as aquatic as Hesperornis, but they were semi-aquatic. And those ones are more, what I'm seeing from their skeletons is they're more like the pelican. They don't have particularly reduced skeletal pneumaticity. And we do see evidence of not only paramedullary diverticula in Ichthyornis and Janornis, but actually really big ones. So that was super interesting.
And then along those lines, then, what would you say about bald eagles and ospreys with similar adaptations, seeing that they're mostly seagoing hunters?
Yeah, I would say that in the case of these structures, paramedullary diverticula, that they're not, I honestly don't think they're adaptive, except maybe in really extreme cases, like what we see in the pelican, where they're completely wrapping the spinal cord, and it's like that throughout the whole vertebral column. I just am not sure that they are adaptive. We do know that the way this pneumatic or the respiratory system develops with the air sacs and the diverticula, the small extensions from them, to a degree, they kind of just keep going wherever they can. They grow and they make holes in bones, and they go as far as the body will allow. So I think my personal guess right now is that that's what's happening. And since it's not adaptive, that's why we see some pursuit divers have them and some don't. And I think it's more determined by the developmental program of these different large groups. And those that they just have them or they don't have them.
So you answered, I think, the next question, which is, what makes the pelican weird compared to the other birds you studied?
Yeah, the pelican just has crazy, their skeletons are highly pneumatized. And then we saw, like I said, and showed the really, really big, super weird paramedullary diverticula surrounding the spinal cord. Just like totally, to me, totally nuts and crazy that part of the respiratory system is doing that.
And then along those lines, you mentioned, so the question was, could lifestyle of birds be a factor of variation?
Yeah, that is a really, really good question. And it's one that I would say we need to test it more rigorously. But based on our preliminary data, I don't think it doesn't seem like it does.
Wow. So it's a phylogenetic roll of the dice?
Yeah, I think so. Mm-hmm.
And then could flight be a factor, like whether they fly or become flightless, and which flyers use thermals or not? Or is this independent of the paramedullary?
I would say this is independent, because we saw some of the biggest and most pronounced paramedullary airways in ostriches, which definitely don't fly. Although I would love to see like a penguin, something else that doesn't fly, but in a completely different way, like a modern swimming bird. Although they definitely have highly, highly reduced pneumaticity. So now that I say that, I bet you anything they don't have paramedullary diverticula.
That was one of my questions was penguins, so you got to it.
Does flight style correlate with this pattern, or is further testing needed?
I think that needs to be tested further. Our first step with this project was just kind of to get a general sense of who has them, who doesn't, what do they look like. What we did not do yet is drill down into those more refined differences in flight style. And I think it's a really cool idea. Yeah.
Could lifestyle factors such as diet and habitat be used to study variation?
Yes, absolutely. There is totally potential for that. I still am inclined to think that perhaps these structures aren't adaptive because some of the variation we see is not just between different birds, but birds with different lifestyles or diets. Like some of the variation is just within a single type of bird, right? Like we saw weirdness, different variation within just ostriches. But at the same time, absolutely, that is a totally testable hypothesis, and something else for the future for sure. And also, I think in addition to that, so we collected data from as many birds as we had easy access to, but it would be really cool to be able to collect data from more birds, especially more birds within the same family. So test what's going on within each family, and look at differences in a more detailed level like that. And as potentially correlates with diet or locomotion, things like that.
Does air sac anatomy vary by sex?
That's a great question and good idea, and always an important variable to consider. We did know the biological sex of these animals. I had data on that, and we did not notice any differences that were correlated with that.
Do we have any data on asdarchids?
Oh, no, not yet. You know what? This research, so what I have learned from my research with Dr. Wadel, with this grand theme of looking at weird things in the vertebral canal, the reason it gets me excited is because nobody else looks there. Nobody looks in the vertebral canal. Half the time when you go into a museum and you look at a vertebra, there's still matrix inside the vertebral canal. It's not even prepped out yet. So what we're trying to do is spread the word that there's cool stuff to see there, because we want more people to be looking inside the vertebral canal. So I think in so many, I would love just for like all vertebrate paleontologists who study many different groups to be looking in the vertebral canals of different pterosaurs and different theropods and sauropods and looking for evidence of these structures. So I would guess there's a very high likelihood that they would be present in asdarchids. I have not had a chance to look in any prepped out vertebral canals yet, though, to find that evidence. But I think I would love to be able to do that. I think there's a really good chance of finding them there.
And that's a nice segue to this question. I'm aware there were some differences between ornithischian dinosaurs and saurischian dinosaurs. Has there been any detailed research done on this so far, especially on the pneumatic side?
That's a good question. To my knowledge, so I'm going to caveat this and say I'm coming at this from the bird side, and what I know about pneumaticity in dinosaurs is what I have learned from my colleague, Dr. Wadel, he's bringing more of the expertise from that perspective. So my understanding is we so far only have evidence for pneumatic structures in theropods and sauropods. Not in any ornithischians. Dr. Curtis, do you know hasn't been changes in that, have there?
Yeah, so first it's weird to hear Dr. Curtis, but it's always weird. But when I was in South Africa kicking around, I was looking at what was used to be called prosauropods as well. I went to look at Volcanodon and South. And I found pneumatic characters, which subsequently folks found my little post-it notes and were able to make lots of publications on in the early aughts on pneumatic characters in these otherwise apneumatic taxa. So I have often wondered how far down the phylogeny that air sacs go. When did it split? Is this one of those fundamental split characters, or did it derive multiple times? Do you have any sense on it's one and done, or it's happened a few times evolutionarily?
Oh man, that is such a great question. I mean, it's always, when we see a family tree of phylogeny with animals that have the same characteristics spread across it, but it's not present in maybe all of the groups. Did it evolve separately multiple times or only one time and then some groups lost it? I definitely don't consider myself of an expert on the evolution of pneumaticity in dinosaurs or in archosaurs to be able to say. My sense is, I would say I think it evolved once and then we've seen a few losses. But I don't really have any good evidence to back that up. That's just kind of my general Gestalt sense.
An order of housekeeping. We're coming up at the top of the hour. I want to be respectful of everyone's time. So do you have a few more minutes to chat with us?
Yes, I definitely do. No
Okay.
problem.
Secondly, I want to bring a few things, kind of pull all of this back together in like a meta take. One, the humility of the researcher here. Dr. Atterholt has repeatedly, I don't know, I haven't touched it directly. This is a classic scientist who is making statements about the information that they can put their hands on and show you why, and is not making that extrapolative leap into wild behaviors, etc. So it's always interesting to contrast guest styles and speculation. And the majority of our guests are very conservative speakers. It's fascinating to me to see. But part two, this is work on extant animals. These are animals that are alive. I can look out my window and see birds today, and yet we don't have these answers. And Dr. Whitmer made this comment when he presented. So many times in his lab, they've went to pull off the shelf and check out Biology Today and look at the moose or the rhino and discovered no one's asked the question, or the answer's known, but it's never been written down because it's just, I can see it. So your work fascinates me because you're doing this work that on some levels is so fundamental, and how come it's not done yet? So thank you. And the other piece is I'm one of those guys who has been looking at vertebral columns and neural canals since 94. And I have seen, and I have my notes, little pointy things in certain taxa, and I've wondered, are these significant? And I realize now, after starting to read your work, that there's so much variation within pneumaticity that a lot of these characters that the cladists hang their hats on and they put presence or absence aren't actual phylogenetic signal. They are actually variable characters manifesting on the bone from something they all had. And yet we score them, and then it impacts our cladogram and our relationships and how we think they flow together. And I saw
this topic I do have very strong feelings on. And from what I've seen, pneumaticity in terms of how it manifests on the skeleton, a fossa, a foramina, it's so variable. It can be asymmetrical bilaterally in the same animal. I've seen that a million times. I know you've seen looking at vertebrae, and Matt and I have seen it, where you'll have a few vertebrae with pneumatic structures and then a few with none, and then a few more further down the line that have it. It's like, so with very few exceptions of extremely consistent pneumatic structures, like it drives me nuts using pneumatic characteristics. And also what's really cool about it is sometimes a bone can be pneumatized through a pneumatopore. Basically, that just means a teeny tiny itty bitty little pneumatic foramen that you might not even see from the outside of the bone. But then if you cut the bone in half, or if you CT scan it, you say, oh my gosh, this bone is pneumatic and I didn't even know. So yeah, just like for so many reasons.
I love it.
Did skeletal pneumaticity have a single evolutionary origin, or did it evolve independently in multiple lineages?
I think it's all cool and super weird. And when things get messy like that in science, sometimes on one hand it's frustrating, but on the other hand, to me, it just makes it more interesting and fun. And yeah, I think I would say I'm inclined to say that we have one evolution of air sacs and pneumaticity and a static lung at the somewhere around the base of maybe ornithodira or of ornithodira. And then I think that we just see a weird loss and modification of that in certain ornithischians. And that's really the conservative side of my scientific mind saying that, right? Like, hi, kitty. We, as scientists, we like to think that we're driven by an understanding of the principle of parsimony. The simplest explanation is best. So I'm really using that here to make my educated guess that the loss within one group, right, or ornithischians, it would be only one loss versus 3 separate gains or 2 separate gains if we have it evolving separately in pterosaurs and then dinosaurs, or pterosaurs and only certain groups of dinosaurs. So it starts to get more complicated. But we also know that the biological world is complicated and messy. So it's possible. And I just can't wait to see where the future research goes on this.
And to that end, another great segue, or maybe it's your host. We were told anatomists are found in med school. Let me restate it. Have you ever been involved in medical school throughout your paleontological career? We were told before some anatomists are found in medical school.
Yes. The answer is yes, a hard yes. I actually would advise, if you're interested in a career in paleontology, seek out every and all opportunities to learn and teach human anatomy or veterinary anatomy, because that's how you get a job. Because it's really hard to get a job as a paleontologist, not to be a downer, but it's the reality of the situation. It's very hard to get a job. But whether you focus on the geology or the biology side of paleontology, we all have to know some anatomy. And if you find, like myself, that you really love it, then take it and run with it, make it your specialty and your strength. So yes, I work in a medical school. I work in a medical school with an anatomy department where 3 of us are paleontologists and the other 4 of my colleagues are paleoanthropologists. So that's like and I have many colleagues across the country who work in different medical schools at different universities. It's a great way to get a job and stay employed. And personally, I think it's an amazing way to maintain and hone your skills as an anatomist. I dissecting is one of my favorite things in the world. And studying and exploring anatomy, anatomical variation. So human anatomy is a great option, but also anatomists are needed in veterinary schools too. So veterinary anatomy, dogs, cats, horses, that sort of thing is also valuable and needed.
Did you know Neil Shubin?
I'm trying to think if I ever met Neil in person. I don't think I did. Although when I was an undergraduate at Penn, I had a work study position at the Academy of Natural Sciences where I worked with his collaborator, Ted Deschler. So I know Ted Deschler super well. I saw him like once a week, every day for 4 years. And he has been a really wonderful colleague and supporter of mine. But I don't think I ever got to meet Neil, but Ted, yes. And I also got to hang out and see the real Tiktaalik specimen like all the time, which was really, really cool.
Given my existing background, which field should I focus on to advance my paleontology career?
That is all awesome. I think it sounds like you're off to a strong start there. I would say seek out opportunities. So again, it kind of depends a little bit. To be a paleontologist, we have to understand biology and geology. And so some paleontologists really lean hard into the geology stuff. Some lean hard into the biology side. So broadly speaking, just taking as many geology classes as you can is really good. And biology classes are really good. Now, speaking to my own personal bias, I think that seeking out courses in any kind of anatomy, human anatomy, comparative anatomy stuff would be really, really helpful and valuable to you at this point in time. What else? Anatomy and lots of geology would probably be a really good foundation for you. Physiology, comparative physiology, that would also be good. And then, so in terms of education, that would be classes to try to take that would be good. And then you probably already know from stuff Brian has told you or from learning from our other speakers, but on the whole, I think the vertebrate paleontology community is really welcoming and supportive of students. So start networking already if you haven't already. Reach out to people. If you have the opportunity to attend a conference that's local to you, do it. Reach out to paleontologists who are local to you. I would put you in touch with Peter, but Peter actually has retired now, so I'm not sure how helpful he would be. But yeah, reaching out to paleontologists locally, seeking out volunteer opportunities, attending little conferences and symposia, telling people of your interest, seeking out field opportunities. And that's just, the networking comes in handy so much for all of those things. So a lot of your education experience you can get just from doing that.
Fantastic. We have one question left in the chat, and that is, have you ever used anatomy with 3D modeling or reconstruction of what a dead animal would have looked like?
Oh, yes, I have. Well, an extinct one, actually. No, just kidding. I haven't. So I have all of my CT scans that I work with, but the CT data that I've worked with thus far is all of living animals. And so I have worked on recreating 3D models from CT scans of some of their anatomy, like sometimes I've in the past done a study of skull shape change through development in modern birds. And I'm currently working on creating 3D models of paramedullary airways. All of that's in modern birds, though. So I haven't had the chance to do it in something extinct, but that's something that maybe I'll be working with Dr. Curtis on here soon.
We'll see what we can do. You can join SBP and other paleo societies. There's a virtual conference again this year. Funny side note, Ray Wilhite and I, we said, hey, we should publish on this Brachiosaurus paper. And this SVP, I have a booth for fossil crates. So I really didn't know if I would have the time to put a presentation or a poster. So we signed up for virtual, but we missed a key detail. The virtual conference is the week after. So now Ray's able to show up. He didn't know if he could be there. My time's freed up, but we're locked into a virtual. So read the fine print before you sign up for things. That's a painful lesson that I'm sharing with everyone. When I mentioned about how there's a lot of stuff about modern animals we don't know. It's how we can observe living animals and on YouTube and in the wild, and we think we have all the answers because they're just right there. And every one of you on this call can go into paleo and using extant taxa. Christy and I, Dr. Lopez, we spent last week before COVID took Christy down, we were hanging out with binturong, which if you don't know what a binturong is an absolutely amazing arboreal carnivore. And what I noticed was the chevrons in the binturong and in multiple species of armadillo were reminiscent of the diplodocids. And I've already looked at giant anteaters and pangolins and what have you. So I went to look at them, and the best thing to do is observe them in person. And the binturong, it does some cool things with its tail. But the one that shocked me was the giant anteater, or the armadillos, the screaming hairy armadillo, because where the chevrons elongate anteroposteriorly is right where the tail, when it bulls up, it flips in. So it looks like it's using its tail musculature to retract the tail and bring it back. It's at the base of the tail only. The binturong is also at the base of the tail, and it seems to keep its tail straight out at the meaty base, and then has a prehensile tail down the road where you see these in the tamandua, the tree kangaroo. So there's a lot of chevron morphology that has never been worked out. And the Wild Wonders in San Diego had 5 binturong on display that we were able to study for hours and hours and play with them. And it was an incredibly cool experience. So if you're looking for something fun, Jessie, I got the hookup for Wild Wonders to go down and spend a day. Just let me know.
Oh my God, yes, please.
Okay.
I go to San Diego all the time. My sister lives there. Yes.
We'll chat offline here in a few moments, and Christy and I can share those details. Then the last piece is I just want to say thank you. You had an owl chick, and it had lots of little feathery. Were those the feathers of the baby owl?
Yeah.
Okay.
Yeah.
That's so cool. It was just like this little puff ball. It made me smile.
Can you discuss sexual dimorphism?
I think that we definitely do have at this point in time, pretty good evidence of sexual dimorphism for at least some enantiornithines, because there are some with just those crazy, huge, weird, truly ornamental tails. And if we have sexual selection, then, it's likely that that's an indicator of female choice, but the scientist part of my brain is also saying we actually, without further evidence, although at this day and age with osteohistological studies and the potential for finding medullary bone, we could potentially sex them. At this point in time, that research hasn't been done, so we don't know for sure that the ones with the ornamental tails are females and not males. So I would say that there is some sort of mate choice, evidence of mate choice going on, absolutely, and evidence, yes, therefore that the idea of good genes, and the females or potentially the males seeking those out is really important. So I think we see good evidence for that in some Mesozoic birds like enantiornithines, and that's part of our phylogenetic bracket, of course, for theropod dinosaurs. So that's getting into territory that I am a lot less comfortable with. But I would say, as a biologist generally, I would not be surprised at all if we in the future do see really good evidence of sexual dimorphism and mate choice in theropod dinosaurs or other dinosaurs for that matter.
So thank you all so much. Dr. Atterholt, tremendous pleasure having you. And if you can stay tight for a moment to finalize some items, the rest of you, good luck, good hunting, fly, be free, and we'll see you in 2 weeks. Thank you kindly. Adios.
Dr. Brian Curtis is a dinosaur paleontologist best known for his research on sauropods, the long-necked giants of the dinosaur world.
This animal is near and dear to my heart. This will probably be the name-bearing specimen for our new species of Diplodocus. And I dug this out. I'm not even humble bragging, I'm bragging. This took a long time to get out of the ground.
He has collaborated with scores of the world's leading paleontologists and has worked in hundreds of natural history, science, and dinosaur museums around the world.